Hi, welcome to this particular module, and here we will be discussing about what do you mean by bacterial antibiotic resistance. Now, this is a extremely hot area of research, since the effectiveness of the antibiotics on killing the bacteria is getting lower and lower day by day. The reason being that we take lot of antibiotics and the bacteria starts resisting to the antibiotic. It cannot be killed by a particular antibiotic. So, why we need to design an engineering platform for understanding whether the bacteria is resistant to antibiotic or not. There are two cases I will discuss. First case is urinary tract infection, and then we will take out of two cases, one case we will take and we will see how we can use our engineering approach to solve this particular problem. So, first is urinary tract infection. Urinary tract infection occurs because of the bacteria called E. coli, and the concentration of the bacteria E. coli is about 10⁵ colony forming units per ml. Now, this causes the infection, and generally when a person suffers from a infection, a bacterial infection, he or she is prescribed topical antibiotics. So, what do we understand by topical antibiotics? Topical antibiotics are 5 different antibiotics that is given, and the samples are taken, whether it is a urine sample, and send it to a path lab. In the path lab, they grow the bacteria on the using the growth media on the disk, and then they use the antibiotic capsules or discs on this particular plate, and if the antibiotic is effective, the bacteria surrounding the antibiotic will die. Thus, what we see, if the antibiotic is effective, then the clear area would be bigger or the radius would be larger, right. I will show it to you what I mean. And the whole process of growing the bacteria, that is first capturing the bacteria, and then to test several antibiotics, 4-5 antibiotics, takes about 48 hours. That varies on the type of bacteria you are testing. So, once the report comes that out of 5, a particular antibiotic would be more effective for a particular bacteria, that is E. coli. Then after 2 days or 3 days, a patient is called back, and out of 5 antibiotics, only 1 antibiotic is he or she is asked to continue, right. So, where is the problem? The problem is that we do not have any engineering device that can rapidly capture and understand whether the captured bacteria are resistant to antibiotics or not rapidly. Now, why to worry? Let us say if I have a bacterial infection, if I go to a doctor, I can stay for 2 days with the 5 antibiotics. Okay. Third day I can get 1 and I can wait for 2 days. But when we talk about neonates, neonates are 1 year, 2 year old babies. their immune system is not as strong as ours, and waiting for about 2 days or 24 to 48 hours or more is not okay, because they die. And this bacterial infection that occurs in babies is also called sepsis, SEPSIS, sepsis. So, in the blood other than E. coli, there is an urine in blood, there can be other bacteria such as staph, staphylococcus, there can be MRSA. So, the point is, can we develop a technology that can on chip capture the bacteria and understand the antibiotic resistance using electrical sensing rapidly, right. That is our given problem. So, okay, one way of doing it is a static way, and second way of doing it is using macrofluidic. Here it is not about flowing the sample like all our other topics like immunotherapy or chemotherapy or drug screening device, right. We will talk about flowing the sample in the macrofluidic device. Here we are not worried about flowing the samples, here we want to test the samples quickly. So, how it will work, let us see in today's lecture. So, when you can, if you see the slide, what you see, the most common UTIs, urinary tract infections, occur mainly in women or women and affect the bladder and urethra. Infection of the bladder, that is cystitis. This type of UTI is usually caused by E. coli, a type of bacteria commonly found in the gastrointestinal or GI tract. Okay. So, you can see bacteria travel up the urethra, you can see here, right, the green color one. This is bacteria from skin or rectum, from this area it can travel, you can see, right, and it travels all the way to the bladder. So, how to know whether a woman is suffering from UTI or not, that is the problem. Here what you can see is how the current gold standard techniques are used. This is a disc diffusion technique, is also called antibiogram assay, and here we have what you can see here is ampicillin, which is one of the antibiotic for E. coli. We have used different concentration 0.1 gram, 1 gram, 10 microgram and 100 microgram. Again 0.1 microgram, 1 microgram, 10 microgram and 100 microgram, and you can clearly see that the bacteria here has died, right, means 10 microgram is more effective compared to 100 microgram is more effective compared to 10 microgram, compared to 1 microgram and compared to 0.1 microgram, right. But there is something called minimum inhibitory concentration, MIC, that one has to take care when prescribing the antibiotic, alright. So, point is, this is how it is done, like you can see here very clearly, right, the effect of the disc diffusion technique, and everywhere there is a bacteria which is your E. coli, alright. So, for this what we can do, what I was talking about is, so when the sample is collected, it is first grown, it is from the sample, the bacteria from the sample is taken, is extracted, right, or is captured and is grown on the using the growth media, right. Then on this sample, once the bacteria is grown, we load four different antibiotics. If there are 5, there can be 5 antibiotics, right. Now, if the antibiotic is effective, then what will happen? Then the diameter will increase. So, that is what we have seen here. You can see this particular diagram. What do you see? The diameter has been increased, right. Now, how we can perform the experiment? How we can come up with a technology that can be faster, that is rapid, and it should not be costly as well, all right. So, if you see this particular device, and then we will see how we can fabricate this device. This device consists of a plastic substrate, which is the substrate on which the device is fabricated. It has an integrated heater, on which there is an insulator, on which there are interdigitated electrodes. Interdigitated electrodes you guys know, right. What is interdigitated electrodes? Electrodes like this, correct, interdigitated electrodes. They are not attached, which are not connected. So, what is it having? It is having a heater, right, it is having a heater, then on the heater there is an insulator, on insulator there are interdigitated electrodes, and there is a contact pad here. This is what you can see here. There is a contact pad for sensor, sensor is nothing but interdigitated electrode, contact pad for heaters, and then on that you have the channel. So, you have a PMMA sheet, PMMA, which is cut, and you create a channel, and you stick the channel on this particular chip, so that there can be an inlet and then can be a outlet. So, what you do with that, right? First is you need to create the micro heater. Now, in this technique, since it is a plastic substrate, a screen printing technique is used. You see, until now what we have learned? We have learned everything can be done using a clean room, a microfabrication can be done when you have a thermal evaporator or a e-beam evaporator or a high end technology equipment like photolithography, or you can talk about RI, right, or you can talk about wet etching. But what if your laboratory, your place does not have these high end technologies? Can you still do research, right, without these technologies? So, how can we design a sensor in which even we are not surrounded by, where we do not have the access to a set of technologies, can we come up with a novel solution? So, in this case what I am showing it to you right now is a device that is made using a technique called screen printing. So, we can screen print a heater, right, everybody knows screen printing. On that, so can you have an insulator on the heater, because you do not have a technique for depositing insulating material like PECVD or like sputtering. You do not have any technique to deposit an insulating layer on the heater. So, what you can do? If you do not have this kind of technique, what you can do, right. So, I will show it to you. Now, let us consider this is a plastic substrate OHP sheet. Okay. On one side I have a heater. I do not have a technique that I can grow insulator on the top of the heater. What I will do, on the bottom side I will use the electrodes. So, now my this hand, which is my left hand, right, is the electrode on the substrate, and my right hand is my heater. Okay. So, you do not have to worry about the insulating layer, because the substrate itself is an insulator, is a plastic sheet. And then on interdigitated electrodes, which is my left hand, you have the channel. So, you can pass a liquid through the channel. This channel is made out of PMMA, alright, acrylic sheet PMMA. And how can you cut the channel? You can cut the channel with the help of laser cutting. So, these things you can have a sensor, you can easily have a sensor. Screen printing on one side of the substrate, heater, screen printing on another side of the substrate, interdigital electrodes, and then on interdigital electrodes you have the channels formed out of PMMA. Now, what you will do? So, if you see here on the slide, we talked about this, right, a chip in which one side there is of OHP sheet, there is a heater, on another side there is a sensor or interdigitated electrodes, you can see here, right, and these are nothing but, when you say contact pads for sensor, is nothing but contact pad for interdigitated electrodes, and you have your microfluidic channel. This is the actual photograph, this is the actual photograph of the device, and how the experiment is done, right, how the experiment is done. You see here white blood cells, RBCs, bacteria, antibiotics and antibody, you should remember this. Okay. This design is white blood cells, which is a gear like design, and it is not actual representation how the white blood cell looks like, it is just a schematic representation to distinguish between the RBCs, WBCs, bacteria, antibiotics and antibody. Okay. So, now what we will do is we will spike the whole blood. So, we will take a blood and we will spike the blood with a known concentration of bacteria, let us say 10⁵ CFU per ml. I know what is the concentration of bacteria in my sample. So, that is called spiked up, right, known concentration adding in the blood is a spiked blood. So, what we will do, we want to first on a interdigital electrodes, you can see here, right, there are antibodies, right, antibodies which are immobilized antibodies, they cannot move, immobilized antibody, alright. So, antibodies are like this, of course, it is like this actually, right, immobilized antibody, and once we have immobilized antibody, we will load spike whole blood onto the channel. So, it will look like the one in this particular figure. You got it? First step is to to immobilize your antibodies, means you have to load the antibodies in the channel and incubate it for half an hour, so that it will be immobilized. And here we are not performing any particular surface chemistry or surface modification. So, our sensing would be the antibodies will be stick to the substrate using affinity. So, this is called affinity based sensing, okay, affinity based sensing. So, now you have the antibodies loaded on the chip. What you will do? You load the whole blood, onto the channel like you can see here in the schematic. After that you have to incubate for 30 minutes and then wash with PBS. When you wash with PBS, what will happen? The bacteria in the blood will get captured on the antibodies. the bacteria that are present in the blood will get captured on the antibodies. Next step, what is the next step? Next step is, once the bacteria are captured, you load antibiotics, which is here, right. You load antibiotics with growth media plus antibiotics. When you do that, what will happen? There is a growth media and antibiotics. What is the role of growth media? Growth media will increase the bacteria. It will help the bacteria to grow, growth media, right. Role of antibiotics, antibiotics is supposed to kill the bacteria, right. Antibiotics are supposed to kill the bacteria. Right. So, if the antibiotic is not effective, either the bacteria will start growing or it will be stagnant, it will not die, it will not grow. This is the case when antibiotic is not effective. You got it? If the antibiotic is effective, the bacteria will start dying. In the presence of growth media, if the antibiotic is effective, then bacteria would start dying. Now, whether bacteria will grow or it will remain as it is or it will die, this we can measure with the help of interdigitated electrodes. And how? By using the impedance analyzer. So, we can measure impedance like you can see here, right, of the interdigital electrodes when the bacteria are incubated with antibiotics in presence of growth media, right. So, what you see here, there is a heater voltage and then there is a impedance measurement. Now, why there is a heater? There is a heater because we need to keep the bacteria at 37 degree centigrade. You have to culture the bacteria at 37 degree centigrade. So, if you see figure 1, if you see this figure is a experimental design schematic. A is a schematic, right, diagram of biosensor with microfluidic channel, B is photograph of biosensor with interdigital electrodes, PMM microfluidic channel, integrated heater and temperature sensor. C will be antibiotic susceptibility measurement using label free electrical sensing on biosensor without surface modification. See, label free, that is very important, right. We are not using any labeling techniques to or we are not staining the cells. So, it is a label free technique. D would be, D is this one, right. On chip bacteria capture and isolation from whole blood using affinity based sensing and measurement of bacterial antibiotic susceptibility using electrical sensing, correct. So, this is one way of testing the, first is capturing the bacteria and then testing the bacterial antibiotic susceptibility. So, how to create this kind of chip? How to create this kind of chip? Like I said, if you have a substrate which is plastic substrate, you screen print heater on one side of substrate and then you reverse the substrate, right. If you reverse the substrate, substrate will look like, correct. Now, on the top you screen print interdigitated electrodes, correct. What you have done? First step is you screen print heater on the plastic substrate, then you rotate the substrate, turn substrate 180 degree and you screen print interdigitated electrodes. On that you create a channel, the cross sectional view, right. So, how you can create channel? You create a channel in PMMA sheet like this, right. So, this is a channel that is created, and then you load the you first load the antibodies, then you load the blood, then you capture the bacteria, right. After you load the blood, you have to incubate it, wash it with PBS, capture the bacteria, then add antibiotic with growth media and look at the change in the electrical property, that is your impedance. So, the electrical characteristic of a screen printed heater on a flexible plastic substrate is shown here. You can see here, as you increase the voltage, the temperature of the heater also increases and the R square value is close to 0.94, right. So, this plot shows the A shows a linear relationship between applied voltage and temperature with R square of 0.954 in an empty micro channel. What does B shows? B shows a plot which shows the response of the heater for different voltages, right. You can see the response of the heater for different voltages here. Now, whether your heater is stable with respect to time for a particular temperature. So, that is what your C plot shows, that the temperature stability of the heater for 14 hours at a constant voltage of 9 volts in an empty channel. Error bars, these are error bars here, you can see in the red color, right, represent standard error of mean. For n equals to 3, we have performed 3 time experiment and p value is less than 0.005. So, we have performed the characterization of the screen printed heater. But like I said, if you want to test multiple antibiotics, you need to have a microfluidic technology. For testing 3 different antibiotics, let us say 3 different antibiotics at a time, you require microfluidics or microfluidic chip. 3 antibiotics at the same time for one bacteria, one particular bacteria, or 3 bacteria you capture, 3 different bacteria you can capture and test 3 different antibiotics and test antibiotics for individual bacteria, right. Let us say there is a bacteria A, B and C that you capture, you can test antibiotic for A, antibiotic for B, antibiotic for C at the same time using microfluidic chip. So, what do I mean by that? Let us see. So, a microfluidic chip for rapid bacterial antibiotic susceptibility testing, you can see here, this is a channel, right, this is a channel, this is one straight and one goes like this, correct, channels are there. Now, what you see here, there is a loading chamber, right, and this is the chamber here, here and here, and below that there is a micro heater, at the bottom there is a micro heater, which you cannot see in this one, but there is a micro heater. You can see here contact pads for heater. So, and on the top we have designed the micro channels with the help of PDMS. So, we have performed soft lithography, we have performed a soft lithography. So, you can see the device here, and this device, this is the outlet, this is the inlet, this is for the or you can say this is the inlet, let us say, because we have the design in this particular format, right. This is the outlet and we have here wash buffer, right. These three things 1, 2 and 3 are for loading antibodies and antibiotics with growth media, all right. These 3 points or these 3 inlets are for either antibodies or antibiotics plus growth media. First you have to load antibodies and then you have to go for antibiotics plus growth media. So, how does it work? How does it work? First let us see how does it look like. I have the chip in my hand. So, if you can just look at my hand and you can see the device, I will show you the device. If you can see the device in my hand, right, this is a device right now, and what you can see here, let me just put it here, yeah, this is better. Okay. So, what you can see here is the chip that I have, right, and I can just show you in this particular format, because I want to show you the PDMS layer, right, this PDMS, and then you can see a channel, right, you can see a channel, you can see interdigital electrodes, and there is a heater. There is a heater here, there is a heater, and this everything is on the front side, everything is on the front side. The reason is that because we need to have, we can have a insulating material on the heater, right. For the device that I am holding, there is a heater on which there is an insulator, on which there are interdigital electrodes, and then we have the channels formed in PDMS, and we have attached this PDMS onto this glass substrate. And what are the channels in PDMS? What is the design? We have just seen in the slide, right. So, this is the device. Now, let us see how we can fabricate this device, and then we will see how we can test this device. So, let us see how we can fabricate. I will show it to you here. So, the fabrication, first thing that we have to do is to design and fabricate a heater, right. So, there is a glass substrate. So, we start with a glass substrate. On that glass substrate, we will deposit a metal for micro heater, right, metal for micro heater. On this, we will perform photolithography. Now, I am not going into depth of photolithography, you guys know how to perform photolithography, right. So, when you perform photolithography, you will have heater, correct. You can have a micro heater. Next step would be to deposit silicon dioxide on the micro heater. After we deposit silicon dioxide, what we will do? We will remove silicon dioxide from the windows for the contact, and remaining area would have silicon dioxide. So, what you can see that there is a micro heater and there is a silicon dioxide, and this is also silicon dioxide. You can say it is a glass, right. Now, on this what we will have? If you just see let us see the screen please. So, after this what we will have, we will deposit a metal. On this we will deposit a metal as I have drawn here, and then we will perform again photolithography. So, when you perform photolithography, what we will have? We will have heater, we have insulator, contact, heater coils, interdigitated electrodes, right, and this is our insulator, correct. So, what we have? We have interdigitated electrodes, we have a micro heater, right. I have written here micro heater as mh, let us use a micro heater like this, and we have silicon dioxide, correct. Now, what we will do? Now, we have this particular substrate, right, we have this substrate. So, this substrate is having a micro heater, it is having an interdigitated electrode and a insulating layer separating the interdigitated electrodes and the micro heater. Now, we want to create a channel in PDMS. What I will do? I will take a silicon wafer, right, grow oxide, then I will perform photolithography, so that I can etch silicon dioxide from the unwanted region, right. Then I will perform deep reactive ion etching, DRIE. When I perform DRIE, what will happen? The silicon in the area which is not protected by oxide will stay, and the area which is protected by not protected by oxide will get etched. The silicon in the area which is protected by oxide will stay, and the area which is not protected by oxide will get etched, right. Now, I will remove silicon dioxide. How can I etch silicon dioxide? I can etch silicon dioxide by dipping the wafer in buffer hydrofluoric acid. On this I will pour PDMS, on this I will pour PDMS and then cure it. After pouring PDMS, we will cure it. After curing, we will strip it off. When I strip it off, this is my PDMS, right. If I strip this PDMS off the silicon after curing, I will have this PDMS. This PDMS I will take and I will just load it right over here. Right, or you can, if you want to have like this, you can light over here. correct. This is what? This is your PDMS. This makes your microfluidic chip. This makes your microfluidic chip. You got it, right. First step is a heater, next step is an insulator, next step is remove the contact from the remove the silicon dioxide from the contact area of the micro heater. Next step would be deposit metal for interdigital electrodes. Next step would be to pattern the interdigital electrodes. On that we will create a silicon mold and we will pour PDMS, cure PDMS, strip off PDMS and then bond PDMS with glass which is having an interdigital electrodes and a micro heater separated by an insulator using oxygen plasma bonding, So, to have a device which you can see right over here, and this is a schematic represent that the cartridge is there in which your device is placed. Within this cartridge consists of your antibiotic 1, 2, 3, waste reservoir, blood media and wash buffer. There is a contact pad for impedance measurement, there are contact pads for applying voltage to the heater. So, how it will work? We will see here, there is a heater voltage, you can see here in our device, because the device that I have shown the heater pads and the impedance measurement pads are on the same side. So, there is a heater voltage that we can apply, then it will be 37 degree centigrade, right. These are capture chambers, you can see 1, 2 and 3, and this is the these are electrodes for impedance measurement. If you see in this particular case, which is your B case, the first we will load in each chamber 1, 2 and 3, each chamber will load antibodies for E. coli. So, it is anti-E. coli, antibodies for E. coli. Okay. In each chamber we will load antibodies. After loading antibodies, what we will do? We will load a sample at the inlet of the microfluidic chip. So, I will show it to you here, it is easy to see here in this diagram, right. You assume that the chamber has the immobilized antibodies, right, which looks like this gray color, which looks like this Y, like this, all right. Then we are loading a sample, it can be your spike blood, it can be urine in the channel, and once the sample is loaded into the channel, it is incubated at 37 degree centigrade for 30 minutes. Okay. After that, we will wash this channel with the help of PBS, right. You see here, wash with PBS. When you wash with PBS, what will happen? That the bacteria present in the sample will get captured on the antibodies, that what you can see here. Bacteria that were present in the sample got captured on the antibodies, right. After it gets captured on the antibody, the next step that we will do is we will load again here in this, again means in this chambers. Okay. We will load growth media plus antibiotic 1, right, growth media plus antibiotic 2, growth media plus antibiotic 3. When we do that, what we will see? We will see that, see, and then we will incubate it for another 30 minutes. See, we can incubate for 1 hour as well, depending on what kind of bacteria we have. Suppose we have E. coli, the doubling time it gets double is 30 minutes. Okay. So, let us say we have incubated our device which is captured bacteria plus antibiotics plus growth media for about 1 hour, and we measure the impedance, live impedance, whenever we from the point we have loaded the antibiotic, right, to the point that 1 hour is over, we continuously monitor the impedance. So, the advantage of microfluidic would be at the same time you can see or understand the effect of 3 different antibiotics, correct. At the same time you can understand the effect of 3 different antibiotics. So, how the data looks like? How the data looks like? So, the data would look like this thing. You can see here we have tested different antibiotics, and you can see the first graph is for ampicillin, right. The second graph is for the ciprofloxacin, and the third graph is for erythromycin, right. 3 graphs are here. If you see A, B and C, A, B and C is tested for ampicillin, the first antibiotic. The next one, which is our E, F and G, this is tested for another antibiotic, and then the last one, which is I, J and K is tested for erythromycin. The D, H and L, these are our antibiogram, right. You have seen this in the first slide, antibiogram assay, correct. So, antibiogram assay you can see here for different concentrations, right, 0.1 microgram, 200 microgram in all 3 cases, and this is our gold standard. We are comparing the results with our gold standard. We have also done the viable cell studies, and whatever impedance we get here, whatever the values we get here, we have normalized it with here, and this is a normalized impedance plotted for E. coli, which is our control sample, and then the E. coli captured with ampicillin of different concentration, and we can see the effect of antibiotics by looking at the impedance change. And we can also correlate it with viable cells here, that this is statistically different. The ampicillin 100 microgram is statistically different, 10 microgram is also statistically different compared to the control, while 0.1 microgram and 1 microgram is not different compared to the control, right. So, what we see is that from this study we can understand what is the minimum concentration of antibiotic that we should prescribe to a patient. Also we can see that using this technique we can quickly correlate with the gold standard, as well as we can rapidly measure the change in impedance as low as 1 hour, as low as or as fast as 1 hour. So, it is really rapid, you can see here, right. So, normalized impedance versus time about 1 hour we can see the change, that this is for 30 minutes, 30 minutes is because the doubling time of the bacteria, right. And if you see and read this figure caption, is the electrical response of E. coli in presence of ampicillin, ciprofloxacin and erythromycin over a period of 1 hour in E. coli in presence of ampicillin, again this one for a period of 1 hour. Real time impedance measurement, so this is just a repeating, just we should delete it, do not worry about it. So, the A is a real time impedance measurement of E. coli in presence of ampicillin, B is a statistical analysis of the effect of ampicillin, and then C would be the viability test. Same thing stands true for E, F and G, I, J and K. The idea is that now we can rapidly diagnose, we can rapidly capture the bacteria, and we can understand whether the bacteria is resistant to antibiotic or not. Also see here, very interesting, we can also form a electronic module, and this electronic module will be integrated with a display, and then there is a port to load this chip inside this whole electronic module, right. This is the module, this is the final device in which there is a display, and then there is a port to load the chip inside this device, and then we have the electronics within this device. So that whenever we understand, whenever we measure the impedance, we can convert this impedance to a display format, whether the antibiotic is effective or not. So, if you see this slide, you can see that if the antibiotic is effective, you will see a significant difference between the normal and the between the control and the actual sample, and the sample that is studied, right guys. So, this is how we can quickly, rapidly measure the effect of antibiotic. We can capture the bacteria in a effective way, and the reason of happening this is because we are using microfluidics. So, micro channels, if you use, you have to cap the bacteria, amount of bacteria that doubles, we can immediately see the change using the electrical sensing. So, can we use this technology, can we use this device for other applications like capturing other bacteria in case of sepsis? This is what we have talked about E. coli, and E. coli is related to UTI. So, for UTI, this can be a potential device that can measure the or capture the bacteria from urine sample in less than 1 hour, and we can measure the effect of antibiotics on those bacteria and prescribe a patient a particular antibiotics rather than prescribing 4 to 5 antibiotics. That is a concept. Okay, this is a concept. These are only initial or preliminary results. This field is open wide. You guys can perform this experiment in a controlled environment along with a person who knows how to culture bacteria. See, bacteria is extremely harmful. Don't mess up with bacteria. In any case, you have to go through a proper training, right, follow the protocol, get the approval, whether it is a biosafety approval required or whether it is a ethical committee approval required, get all the approvals in place, then and then only test your sample. It is very important to understand. Do not just start fabricating a device and start loading the sample, that it does not work in this particular way, because bacteria and cells are enemy. If you have seen an incubator and if you add bacteria in the incubator where the cells are there, cells will die. So, if we get infected with bacteria, it will affect our body, it will start killing the cells. So, it is harmful. Always have a proper precaution, always go through a laboratory protocol before you perform any of this experiment, not only this one, any of the lectures that we have seen, you have to undergo a proper training and then only operate or work in this area of research. Right. The idea is that can you design a device that can be used to understand the antibiotic susceptibility of a bacteria, or you can say a bacterial resistance to a particular antibiotic, and at the same time can you capture it, and at the same time is it rapid, and at the same time does it require a small volume, because now we are talking about microfluidics, right. So, we have to use only 10 to 20 microliter of sample rather than taking 2 to 5 ml of blood, right. So, think about this, learn this, read the lectures once again, and I will see you in the experiment class where you will be shown different equipment, equipment that are used for clinical applications, whether it is an incubator or is a peristatic pump, or there are microscopes, or it is a micromanipulator, right, it is a impedance analyzer, it is a biosafety hood, right, it is a O1, or we can have a small vacuum pump where we can perform the we can perform soft lithography by pouring PDMS on a mold and then stripping it off. So, these things you will be shown in the laboratory that we have here at Electronic Systems Engineering, Indian Institute of Science. So, till then you take care and I will see you later.